el video para que sea YouTube.
Buenos días a todos y todas, gracias por venir al seminario que nos va a dar el profesor Alain Tissier. Él es el jefe del Instituto de Leibniz de Biochemistry en Halle, Alemania. Él trabaja con metabolismo y biología celular, es profesor en esa universidad en Wittenberg, y él trabaja principalmente entendiendo las relaciones metabólicas en las fábricas que son los tricomas, ¿sí? Y trabaja especialmente en los metabolismos secundarios de los tricomas tipo 4, tipo 6. Tiene una lista larga de artículos al respecto. Hizo numerosas contribuciones en entender las vías de biosíntesis de estos compuestos y el rol que tienen entre el metabolismo primario y secundario. Bueno, además está interesado en la ingeniería metabólica y la biología sintética, y también tiene numerosos trabajos al respecto, y hoy nos va a hablar acerca de cómo heat stress and via jasmonate signaling change trichome development and metabolism in tomato. Perdón mi inglés, que parece alemán. Bueno, thank you for coming. And, bueno, esto va a quedar grabado, se va a transmitir en vivo y después pueden quedarse, hacer preguntas y charlar con Alain, que todo lo que ustedes quieran. Muchas gracias.
Thank you very much, Milena, for the introduction. I think you did okay. So yeah, it's really nice to be here in Buenos Aires. This is my first time in South America, actually. So it's really nice to be here. So yeah, so today I will, before I start the actual presentation, since maybe
yes.
I will present my institute briefly. Yes, I think it should be here.
Sorry, if you want, you can keep talking in English.
Yeah, so the institute is based in Halle. Maybe you can move to the next slide. So it's a city which is south of Berlin, and this is in the former East German Republic. Maybe I think it's here, isn't it here?
Yes, but
it's
we
not
don't
started?
see it in the in the presentation. You know what? We'll put this 1 minute. All yours.
Okay. So, yes, so Halle is in the east part of Germany, as shown here. This is part of the former East, so German Democratic Republic, and in Halle the institute is located outside the center on a very nice campus. There's a lot of green area around it, and the campus has a number of institutes from the university, but also outside the university. So this is where we are here, near the river, and there's these are the different faculties of the university. There's the University of Hospital there also with medical research, and there's also a Max Planck Institute here on physics, and also Helmholtz Center on environmental research. So it's a really dynamic campus with a lot of research in different areas. So our institute is part of the Leibniz Association. So in Germany you have universities, and you have 4 different kinds of extra university institutes, so institutes that are independent from the universities. So you have the Max Planck Society, which is very famous for really outstanding research. You have the Helmholtz centers, which are more for large infrastructures, the Fraunhofer, which is more applied research, and the Leibniz institute that do fundamental research, but also with some application. And it's with wide areas, wide topics, wide range of topics from life sciences, economics, humanities. There are also many museums that are part of the Leibniz Association. And the institutes are funded by the federal government, 50%, and 50% by the local states. So Germany is a federal republic. Okay, so in our department, the Leibniz Institute of Plant Biochemistry, we have 4 departments. And what's unique about our institute is we have a chemistry department, which is run currently by Ludger Wessjohan, so which gives us the possibility to have access to synthetic chemistry, but also all instrumentation to do the structural elucidation of compounds, for example. So the 3 biology departments are run by Stefan Haber, who works on molecular signal processing. Tina Romais works on plant pathogen interactions, and my department where we work mostly on plant secondary metabolism. And the topics can be, let's say, have as objective the chemical basis of plant resilience, so with objective to enhance plant health and crop performance, generate value-added plant products from secondary metabolites, but also to identify new bioactive molecules from plants for human health, or also for plants. So it's very much focused on plant small molecules, so whether they are secondary metabolites or specialized metabolites, people say today, but also phytohormones. There's a long tradition of work on research on phytohormones. Jasmonate, for example, was identified in Halle, in this institute. And also, of course, on all the proteins that are involved in the biosynthesis or the perception of these metabolites. So we are quite well equipped with, of course, a lot of plant growing facilities, greenhouses, climate controlled phytochambers. We have also very good microscopy, but really the strength of the institute is for everything that has to do with metabolomics, mass analytics, metabolomics, mass spectrometry. So we have a number of instruments. And also one group, actually from my department, that has also been very important is on synthetic biology, so developing Golden Gate cloning. I don't know if you're familiar with this, but this is a method that really has, for us, revolutionized the way we do the cloning. And then recently we got funding to centralize, because until now all the instruments were distributed into the different departments, to bring everything under one roof. And so this new center for metabolomics and computational biochemistry, where we combine all the analytics, metabolomics, but also computational aspects to analyze the data. So this is ongoing now. We are building up this center. Okay, so now in my department, we have 4 different groups. There's the group of Bettina Hauser. I think Carlos knows her, so she works on jasmonate function and mycorrhiza. Thomas Vogt, who works on phenylpropanoid metabolism. For example, he's been working on the biosynthesis of piperin, the principle of pepper. And Sylvestre, who's the inventor of this Golden Gate cloning and who's doing the synthetic biology. And in my group, as Elena mentioned, we work on trichomes, but in general also on isoprenoid biosynthesis. Okay, so this is a recent picture of members of the department. So now we start with the actual research topic that I will present today for my group. This is about trichomes, glandular trichomes from tomato. So in my group, there are 3 main topics. One is really on trichomes and secretory structures from plants. The other is on isoprenoid metabolism and stress, so for example light stress and heat stress, so there's also connections of course between those two topics. And another aspect is more on metabolic engineering and genome engineering. So, but the topic I will talk about today is really on trichomes and abiotic stress. Yeah. Oops. Something wrong with the pictures. That's okay, we don't need the pictures. So trichomes are hair-like structures that are present on the surface of leaves and the aerial parts of plants. They're present on many plants and they're important for the protection. They constitute a first barrier of defense against insects, pathogens, but also they contribute to the protection against abiotic stress. So for example, they can protect against light, but also heat. You know, many plants that live in arid environments have a high density of trichomes. And they're present on different plant species. Unfortunately, here somehow the pictures are gone, but and they also provide glandular trichomes, so trichomes that produce metabolites, provide interesting compounds that are useful for humans. So, like for example, artemisinin as the anti-malaria compound, one of the famous compounds, or cannabinoids from cannabis that are produced also in trichomes. Okay, so in tomato, which is the topic of today, it's interesting because there's quite a diversity of the types of trichomes that are present on the surface of tomato leaves. Yeah, in contrast to Arabidopsis, which has only one type, tomato has up to 7 different types that are present of different sizes, and also importantly, that some of them are non-glandular, so they're just hairs. So it's more like a physical protection. And also glandular trichomes that have at their tip cells that are specialized for the production of metabolites. So those metabolites can be, for example, volatile compounds like terpenes. And these are essentially produced in the type 6 trichomes shown here, that have 4 glandular cells at the tip, but also acyl sugars, which are the sticky compounds that you find on the surface of the leaves. And these are produced by the type 4 or the type 1 trichomes. And these are particularly important for the protection against herbivores, insects, because they trap the insects. So today I will talk essentially about these 3 types of trichomes, type 4, 5, which is, let's say, the homologue of the type 4 but without the glandular cell, and the type 6 trichomes.
No.
Something wrong, I don't know why, there's a problem with the presentation. Type 4 trichomes, there's a publication from a group in Brazil, from the group of Lazaro Perez, in 2017, where they showed that, so in tomato, the type 4 trichomes, those glandular trichomes in the mature part of the plants are actually absent, or very rare. Yeah? But if you look in the cotyledons, and also a little bit on the leaves, those trichomes basically are present, but they disappear when in the mature leaves. That's what you would see here, and instead the type 5 trichomes are almost absent at the beginning, and then they replace, if you will, the type 4 trichomes in the more mature stages of the plant development. So, yeah. And there are also differences in between tomato species. So Solanum pinili, for example, which is a wild, I don't know when we checked.
I guess it's PowerPoint.
Yeah, but when we checked, it was okay. And,
Yeah.
and now it's, I don't know why. Yeah. Or maybe you can switch to
Maybe you have a PDF.
PDF.
It would work better.
but I would need to transfer it to PDF. Okay, let's see. I don't know if I think the rest is okay. This is just these.
Okay.
Yeah. So also there are differences between cultivated and wild species of tomato. So for example, Penelli, Solanum Penelli, has a lot of type 4 trichomes. It's very high density of type 4 trichomes. So there's really differences between tomato species, how the trichomes are developed. Okay, so in this project, what we've looked at how heat treatment affects both the development of trichomes and the metabolism of trichomes. Yeah? So these are the experimental conditions. So the control plants are grown at 25 degrees during the day, so during the night a little bit cooler. This is the long day conditions. And the heat treatment is after 1 week of germination, so the plants have just the cotyledons and the first leaves. And then for 3 weeks, 37 degrees during the day, and nights are cooler, because if it's constant 37 degrees, the plants really don't like it. You know? So what we observed, which was quite surprising, is that heat stress induces type 4 trichomes. So I told you that type 4 trichomes so when you look at those mature, more advanced stages, leaf stages, there are hardly any type 4 trichomes anymore. So what you see is in green the type 5 trichomes. This is in the control, so you have many more type 5 trichomes compared to the type 4. Again, you see here the glandular versus non-glandular, and then heat stress is basically reversed. The ratio is reversed. Yeah? These are some pictures that illustrate this. So you see here, for example, in the control condition, mostly type 5 trichomes, so the non-glandular type that you can see here. And under heat stress, you see a lot of these these trichomes with the glandular cell at the tip that start to develop. And you can see here, it's interesting to see that the decision to make these glandular trichomes is you can see already they start differentiating this glandular cell at very early stage. So what happens is, it's not that the type 5 trichomes become type 4 trichomes, it's that at the initiation of trichome development, then there's a decision to make one type of trichomes or the other. Yeah? So it's not a switch from type 5 to type 4 when the trichomes are mature, but it's a switch in the initiation of the trichomes. Yeah? So those trichomes, type 4 trichomes I mentioned, produce acid sugars. So acid sugars are molecules with a sugar core, so in this case sucrose, with different short-chain fatty acids that are esterified at different positions. And so the tomato produces a range of these, so different compounds from this class. And the difference in these compounds is shown here in the nomenclature. For example, here S417 means it has 4 fatty acids, in total 17 carbons, and these are the length of the fatty acids. So 2 is acetyl group, and 5, these are fatty acids with only 5 carbon atoms, so relatively short carbon atoms. And so you can measure those by CMS, and you see here the difference between heat stress and control. There's really a massive increase in the production of these acid sugars and the heat stress, and you can see here the difference between control and heat stress. It's really a huge increase, more than 50-fold increase. And what's striking also there, if you remember maybe from the previous slides, the increase in trichomes is maybe, I would say, 4 to 5-fold maximum, yeah? Whereas in the increase in acid sugars is much more. So that means there are at least 2 processes that are at play here. One is the induction of the trichomes, and there's another level that the production of the acid sugars is also induced by heat stress. And we see this, or this is reflected also at the level of gene expression. So I won't go into the detail, but these are genes that are involved in the biosynthesis of acyl sugars. They've been characterized mostly by the group of Rob Last and at Michigan State University. And you see that, I mean, they are all induced to a variable extent, but some genes are really very strongly induced. Okay, so then what I've shown now, until now, was a continuous or a heat stress for 3 weeks, and we looked after 3 weeks of heat stress. Yeah? So the question was, how long should the heat stress be to trigger this switch? Yeah? So we performed experiments where we did a heat stress for 2 days, for 3 days, for 4 days, and then looked also at the end of 3 weeks what happened. And what you see is that 2 days is already enough to induce, 2 days of heat is already enough to induce this switch. And the longer the heat stress is, or 6 days, the stronger, I mean, the more type 4 trichomes you see after this period, and also the more acyl sugars you see. Yeah? But already after 2 days, it's enough to induce this process. So it's a gradual process. Here you see the stages of the leaves after 6 days. We also asked, so here we looked again after 3 weeks, yeah, but we also, if you look directly after that time here, so after 2 days, 3 days, 4 days, 6 days directly there, so here it's expressed as the ratio of type 4 to type 5. You see that there's under heat stress also already a switch in the ratio already after 2 days. And also you see again this increase in acyl sugars, which is detectable very early. So it's a process which is quite fast. You apply the heat stress and it's in a matter of days, it's I mean quite fast. Okay. Now we also ask ourselves what happens, because there's another type of trichome, the type 6 trichomes that produce volatiles, so monoterpenes and sesquiterpenes, and they're shown here. What happens there? So what we saw is that there's basically no change in the density of these trichomes. They don't seem to, there's no induction. What we saw on the other hand is that in some cases, normally those trichomes have 4 cells, and you see that there's here you have additional cells that differentiate. So it seems to have an impact on the development of the trichomes. But what we observe is that monoterpenes, which are actually in cultivated tomato, are the most abundant terpenes, that they are increased by heat stress. So you see here, for example, the most abundant one, which is beta-phenandrine, is increased. I mean, the increase is not massive. Yeah, it's maybe twofold, maximum twofold, but it's definitely there. Yeah. On the other hand, when you look at sesquiterpenes, which is the other class of terpenes that are produced, we see, and they're quite more dramatic, but in this case the opposite. We see a reduction of sesquiterpenes, as you see here. So these are controlled heat stress for the different compounds. For caryophyllene, which is the most abundant, you see here quite strongly reduced. So opposite effects depending, these are all compounds that are produced in the same structures, but heat treatment has opposite effects on these different types of terpenes. So I should say that sesquiterpenes are produced in the cytosol, from the cytosolic pathway, the mevalonate pathway, and the monoterpenes are produced in the plastids, and this is dependent on the non-mevalonate or so-called MEP pathway. And so we looked at gene expression, and here this was quite surprising because we saw that actually for all genes they were downregulated. So even though we had an increase in monoterpene biosynthesis, the gene expression, for example, TPS20, these two genes are involved in monoterpene biosynthesis, their expression is downregulated. And when you look at the precursor pathways, so for example the mevalonate or the MEP pathway, you also see a downregulation in the gene expression. So it's kind of a enigma how although we reduce, so for sesquiterpenes it makes sense because you reduce gene expression, you see less, but for monoterpenes, reduce expression, more production. That's what we see. So, if you know a little bit about trichomes, you know also that jasmonates play an important role in trichome development. There's a number of, quite a lot of literature connecting jasmonate with various aspects of developments in different species, in Arabidopsis, in tomato, in Artemisia, and cotton. So again, there's a number of transcription factors that have been identified. I won't go into the details, but it's just that there's a lot of data that is available on the role of jasmonate, but nothing in the context of, as far as I know, on heat stress. Yeah, so here again the pictures are gone, but this is not so important. So there's one mutant in tomato that has been identified a number of years ago, which is called the JAI1 mutant. This is a mutant that has that is deficient in the perception of jasmonate, and in this mutant, so it's a mutant is female sterile, but this mutant has no trichomes, no type 6 trichomes, so no trichomes on fruits and less type 6 trichomes on young leaves. So it can still make trichomes, but much less than the wild type. But regarding type 4, type 5 trichomes, this has not really been looked at. So we use this mutant, and we looked how this mutant behaves, so with respect to the phenotypes that I just presented, how it behaves under heat. And again, you see the wild type here, so controlled heat stress, this switch in trichome type. The heterozygous mutant has pretty much the same phenotype as the wild type. This is okay. But then, if you look at the mutant, the JAI1 mutant, no change between control and heat stress. So this switch does not occur in the jasmonate mutant. So this shows that basically this process is dependent on jasmonate signaling. And if you look also at the acyl sugars, one has to say the level of production of acyl sugars in the mutant is already quite low compared to the wild type, which is already very low, but there's no induction, which of course makes sense because if the structures to produce it are not there, you cannot make those compounds. Yeah. So really quite clear phenotype. And then, of course, since we saw different effects on type 6 trichomes, we looked at there what's happening, and there the effects were really not really. I mean, there's nothing really dramatic. What was the only thing that was maybe could be seen is that there was less monoterpenes, but again, the problem is you have to see that to start with, those plants have produce less of those volatiles. So it makes the comparison a little bit difficult. So I think the conclusion from this with regards to the volatiles is that under heat stress, there's not much difference between the jasmonate perception mutant and the wild type. So, of course, then one question was, are there changes in jasmonate signaling, so in jasmonate content or in jasmonate signaling? So again, this is the scheme that we used initially, and after 3 weeks, we saw no difference. So when you look at OPDA, so this is a precursor in jasmonate biosynthesis, in JA, so jasmonate or jasmonate isoleucine, there was really no significant differences. This, one has to say, this is without wounding, without induction. So in these conditions, the levels are very low. Yeah? So it's very difficult to see differences because you can see, for example, for jasmonate isoleucine, we are in the range of, it's almost at the limit of detection. So it's hard to see really differences. So, but then since we saw that this phenomenon, this switch, occurs very early, we wonder whether these changes in jasmonate could occur really in the early hours after the application of heat stress. So we did a time course where we looked at when the heat was applied after 15 minutes, 30 minutes, and so on until 6 hours. And there we saw that there was an increase in OPDA, in JA, and also jasmonate, azoleucine. It kind of starts after 15 minutes, but it really picks up after 1 to 2 hours. Yeah. So probably, so this is as the heat is applied, and probably after that there's a kind of adaptation, and the J levels then stabilizes after 1 day. Yeah. And when you look at gene expression, as a control, you see, for example, HSP70, it's a heat stress protein. Of course, it's strongly induced. COI1, maybe a little bit of induction, but this is really at the limit of significance. MIG2 a little bit at the end, so not really dramatic, but what was more significant is the induction of JAS, some of the JAS proteins, so genes. This is gene expression, so transcript levels, JAS2, JAS4, and JAS10 not really. And so there seems to be a transcript program that is induced. It's not dramatic. It's not like after wounding, for example. When you do wounding, you release like massive levels of jasmonate. You really have very strong transcription reduction. It's a more subtle phenomenon, but I think it's definitely there. Yeah, so that's basically the end of the story. So this paper is under review currently. It's on bioarchive, so if you're interested, you can have a look at it. And I'd like to, yeah, this is a short summary. Yeah, heat stress initiates a shift from type 5 to type 4 trichomes in cultivated tomato. I'd say sugar production is also induced by heat stress.